Tell us, what is an exosome and why does NurExone believe that this is the drug delivery of the future?
So exosomes are natural nanocarriers that are used in the body as a cell-to-cell communication. You have trillions of exosomes at any moment in your body. And we recently, when I say recently, it was like 10 years ago, found out that they have remarkable abilities to target specific areas in the body that are injured, inflammatory areas, and they can be used as a delivery for other drugs. Since they are so small and they can maneuver between the tissues and between the cell and to target the specific location, we can load them with a specific drug and they can be the next generation of drug delivery to a specific location in the body or in the brain, which is a very big challenge for science and neuroscience and medicine.
So what is it? Fantastic. So these tiny little nanocarriers that you call them, okay, you can actually put some molecular cargo inside of them. And how do they know? How do they guide themselves biologically to the right location where they're needed? How do they just go to the right place?
So it's like a very developed, evolutionary developed envelope, okay? That has its target. Imagine just some kind of a small, I would say, a ball or a small tiny balloon, which they have like receptors on them, okay, all over. Those receptors, they can sense signals of inflammation or of injury that are secreted from the cells in the injury site. Okay, and when they are wandering around the tissue, they can just naturally stick to the cells in this specific location. When we load them in with another molecule or another chemical entity that can help the cell that absorb them, so what is happening is that the exosomes are being uptaken by the cells. They are open as an envelope inside the cell, and then the cell use whatever the exosome carries. So naturally, the exosomes carry biological information that helps the tissue or the cell regenerate or recover. But if you load another molecule with another ability, you get like some kind of a synergistic effect that the cell uses both the capacity of the exosome and both the new compound that you put, the drug or the medicine. So you get like an unbelievable effect.
So they naive or unloaded exosomes. They have the ability. They also have some therapeutic ability. And then when you load and let them carry something to the right site, they have this double whammy that's really incredible.
Exactly. Just one specific very important point.
Sure.
That they have the natural ability to help the tissues recover and the cells recover if they are being collected or secreted from a specific cell types. Not every cells that secrete exosomes have the ability the same ability. We in NurExone use a specific type of cells that secrete, those are mesenchymal stem cells. They have natural ability to help a tissue regenerate. Their exosomes also carry the important information, but it's a basic information. It has a limit. Okay, that's why we are not having a natural recovery all the time in our body, because we do have these exosomes. It's limited. Once you load them with a specific cargo, specific molecule that encourage it or activate another molecular pathway, then you get the synergistic effect, something that the body doesn't know naturally.
And as a neuroscientist, anytime there's tissue damage or whatever, so I have that inflammation that will draw these exosomes to the site that they're needed.
So this is what we found. That's exactly what we found out in our one of our papers during my PhD work. We used exosomes. We labeled them. Okay, we used some kind of imaging technique to follow them in the body and in the brain specifically to a different type of damages, physical damages, stroke. Psychiatric disorders also that involves inflammation in specific areas. And what we found is that the distribution of the exosomes in the brain was exactly compatible to the locations of the injury. Wow. So and it happened all the time. That's how we found out that when you take the right type of exosomes, the mesenchymal stem cells derived exosomes, they have a natural ability to do so. And we got extremely excited over it because it can target one of the biggest challenges of neuroscience and psychiatry and neurology, the problem of targeted drug delivery to the central nervous system.
Can you talk a little bit about how these type of exosome-based therapies will be administered?
So we used intranasal administration.
Okay.
So
Which
which
is
is
sniffing, you just
exactly
put it, yeah. Okay.
exactly. It,
Yeah.
it can also go from intravenous, which is by the blood. They have incredible ability to cross the blood-brain barrier, to go all over the body, to search damaged tissues, and when they don't find any damaged tissue, when we use like totally healthy mice and rats. So the exosomes just traveled and then they went out in the urine. So they've been evacuated. We saw this. We take the exosomes, we grow them outside of the body, we grow the cells outside of the body, we collect them in a specific concentration, and then we give them to the animal, or in the future to the person, by intranasal or non-invasively, basically.
And because they're not cells, I don't have any immune reaction to them, right? They don't
This is
trigger. Yeah.
amazing. Yeah.
Yeah.
One of the challenges of other types of therapies is the response. How would the body respond to a biological entity that he doesn't know that all of a sudden comes to the body in a huge numbers or a huge concentration. Exosomes are are being activated under the radar of the immune system. So they can go around the body without basically encouraging the immune system to detect them or to be overactive, which is very important.
So this is really exciting. So one of the key elements of this new drug delivery system is how you load the molecular cargo onto the exosomes. And NurExone has an exciting new patent called Compositions and Methods for Loading Extracellular Vesicles, which are exosomes. And the patent has been uploaded to the NurExone website. So NurExone produces the exosomes in its own bioreactor, loads them according to the technology we're going to discuss, and synthesizes drugs. Can you explain what's so complicated about loading them?
Yes.
Make it sound so easy, yeah.
So it is very complicated. The first challenge is how do you load a drug into a small nanovesicle biological compound without damaging the compound itself. We don't want to damage the ability of the exosomes, the natural abilities of the exosomes to target the inflammatory tissues. We don't want to damage their ability to be uptaken, to be recognized by the cells in the tissue and then be uptaken by them. So we need to keep them intact. This is the first challenge. The second challenge is the chemistry of it. Exosomes are a natural compounds. They had been through years of, I would say, evolution to exactly not to take anything that is in their environment, not to accidentally put it inside of a cell like a Trojan horse. Okay, so you need to bypass all of this, and it becomes a very big challenge. There are a few methods of loading exosomes, but they are suboptimal. Some of them ruin the integrity of the exosome. Some of them are not so efficient. We came up with an idea, a chemical solution, a biochemical solution, that we will give the exosome some kind of a treat that they would like, and we will use this treat as a Trojan horse to the exosomes themselves, so they will carry it into the cell. And that took a lot of time figuring out the chemistry, how to do it, and the validation that it's really working. It doesn't damage the exosomes. The exosomes likes this trait, and they really carry it into the tissue and the cells in the inflammatory tissue. So that was a big project, and we succeed of showing everything and.
Well, how do you measure a success? How do you so you check the integrity of the exosomes afterwards?
Mm-hmm.
What else is important after you've loaded some sort of a molecular?
So first you need it's a very complex research to I would say prove first of all that the compound that you want to load has been really loaded into the exosomes. Okay, compounds in biology and chemistry have this tendency to aggregate among themselves. Okay, so there are some techniques that you need to do to make sure that what you think you see is what you really see. Okay, that if you are measuring the amount of a material inside of an exosome, so this is exactly what you're measuring, and not measuring the numbers of exosomes and the material outside that just had leftovers. So I would say demands lots of controls. Okay, but eventually you measure success if after all of this control you can take the exosomes, measure how much of the material, like quantify the amount of the material, the amount of I would say RNA or a drug that you wanted to put inside of the purified exosomes, and the biggest challenge is to put it on cell or to give it into a rat, to take the tissue and to see if you have the compound in the inflammatory area inside of the cells, and this is what we do.
So that's what you've checked in the laboratory. You've loaded a certain amount of molecules of a compound onto the exosomes, and then you check inside the actual tissue to make sure that the amount of compound they expect to see is there.
Yeah, the presence of the compound is inside the cells. This is the biggest challenge. Inside the cell, we give it intranasal, for example. We measure we take the tissue, the inflammatory tissue, after the rat is dead or after we sacrifice it, and then we search for the compound that is not naturally occurring it doesn't have it naturally in this tissue, and we need to find it. And a control of it, for example, is to give the same material without the exosomes, and then you wouldn't find it in the tissue.
Wow. And is it in the exosome or on the exosome, or is it a discussion?
It's a discussion.
Okay.
It's a discussion, and some people would say that it's a very important discussion. It's a very important topic if you just put things on the exosomes or inside of them. Our purpose of this delivery is to show that regardless if it's inside or outside, the compound that we want to deliver is safely delivered to the tissue, safely been uptaken by the cells, and the cells can use it. That was the point of it. And this is what we checked. And now we are measuring, again, the biochemistry of it, how many of it inside of the exosome, how many of it outside of the exosome. Those are some kind of validations that we are doing, but the main goal was achieved. We managed to deliver non-invasively a compound to a damaged tissue in the central nervous system using exosomes.
And your loading technique or your loading technology is it's very different than other ones that were available, that were known previously. The one that's described in the patent.
Yes. So we used a specific conjugation with a linker to the molecule itself. We are not touching the exosomes. Okay. We are not manipulating the exosomes. We are not manipulating the cells that the exosomes been secreted from. Our patent basically works on exosomes that have already been purified from intact cells, okay, the natural way of cells. They've been purified, and we have a full control of how would be the efficacy or the amount of the end product, okay, because it's in process. We already have the exosomes. We can manipulate the amount of the material that we used and interact, and just by simple incubation of the exosomes with this molecule. It can be any molecule, we believe. We did it with short interfering RNA, okay? But it can be done with, we believe, proteins, small molecules, wide range of biological materials, okay? And we can control it because we control the amounts, we can control the purification, and we can measure it.
Tell us a little bit more about what is special about your technology. You use some sort of saccharides that are not toxic, and they're something special for the neuron cells.
This is the treat, basically, that we use to lure the exosomes to take the material. So
Okay.
it's different type. It's a saccharide dependent loading capabilities, which means that it can be different saccharides like sucrose, glucose, and here we change the scientific or the known paradigm of how you treat exosomes and how you can load them with a specific molecules. Exosomes are a bilayer membrane like cells. So the idea was, but they are not cells. This is more or less
Okay.
the thing. So we changed the paradigm by using different type of molecules that are not interacting with the I would say biochemistry of the bilayer membrane of the exosomes. They have different pathways to get in, and that was another breakthrough that exosomes that NurExone had.
So your saccharides, what you said initially that your goal was not to structurally affect the integrity of the exosomes. So by discovering this naturally occurring pathway, you're able to penetrate the
Exactly.
exosomes
Naturally, in a way that they like, in a way that doesn't damage them, and in a controllable manner, which is very important to the industry also.
And what you're introducing is also something natural, so we don't have any worry about toxicity.
Exactly, we have saccharides in our body. So.
Okay, they're tasty. Yeah. Okay, and there's no adverse effects whatsoever of the saccharides.
No, the saccharides, if anything, they can help because the cells are also using saccharides as an energy source. So once the molecule in the cell is separated, this is again a theory of us. We didn't check it until the end, but the common sense says that eventually the molecule is being separated from the saccharide because the cells know how to separate. If anything, the cells can use them as another source of energy, which is helpful and not damaging. So we are on the safe side of it.
So it sounds perfect. I mean, it sounds like it
It
has
is.
all the
Thank you.
elements